Here at Miltenyi Biotech, we are concerned about the emerging antibody-related reproducibility crisis. As a supplier of flow cytometry antibodies and reagents, we see it as our responsibility to provide our customers with high-quality products that lead to reproducible results. Therefore, we have taken steps to address this problem by introducing recombinant technology to our antibody production process.
As a company providing antibodies, you have basically to take care of three things. First is the specificity of an antibody. Second is the sensitivity of an antibody in your particular application. And the third thing is reproducibility. And knowing the hybridoma business quite well, we also know the limitations of this technology. It's a brilliant technology, it got the Nobel Prize, but it has certain limitations. With recombinant technology, recombinant expression, things become much more stable. REAfinity is for recombinant engineered antibody. The antibody portfolio exploits recombinant antibody technology. It's engineered towards, for instance, no Fc receptor binding, and we use antibody clones with a high affinity for the target antigen.
REAfinity antibodies are made to meet new reproducibility standards, providing superior lot-to-lot consistency in flow cytometry and high reagent purity. They enable hassle-free analyses due to reduced background signal and allow for straightforward experiment planning with only one universal isotype control.
Our lab focuses on trying to understand how natural killer cells work, and then ultimately trying to implement that knowledge in clinical trials for patients with cancer. I think the whole field is suffering from poor specificity of antibodies. Many antibodies react to multiple targets. There is sort of non-specific binding to Fc receptors on cells that you're trying to stain. We currently have a collaboration with Miltenyi, where we are looking at the specificity of a range of REA-antibodies. Recombinant antibody technologies allows for more predictable production, I guess, of the antibodies, and also by allowing to engineer the antibodies for reduced binding to Fc receptors, for example, you can reduce background noise and non-specific binding, and that's what we have seen in initial testing. So I think that's a development that we are welcoming very much.
The ultimate goal of Miltenyi Biotech is to develop innovative technologies that enables our customers and collaborators who do cutting-edge cellular research to ultimately translate it into cellular therapy approaches.
With the recombinant antibodies, with the REAfinity antibodies, it's easy to transition from using them for research or diagnostic applications or applications in the cell therapy world.
So in this environment, there's no debate about quality. We need to have it, and we need to have this quality to have reproducible, reliable results.
For more information, visit us at miltenyibiotech.com/REAfinity.